So I'll be collaborating with Shuchi Dutta, who's one of the faculty members here in the Protein Data Bank, to talk to you about this as an empowering resource for understanding HIV and AIDS at the molecular level. So our presentation will be provided in 3 parts. We'll very briefly touch on what is the RCSB Research Collaboratory for Structural Bioinformatics Protein Data Bank. Then we'll explain in some detail the way in which structural data has facilitated the discovery of HIV treatments. And then I'll hand over the podium to Shuchi, who will be talking about a new curriculum which has emerged from the outreach efforts that we're making on behalf of the Protein Data Bank for students, educators, and of course the members of the general public. So let me start with what is the Protein Data Bank. So this is the first open access digital resource in all of biology. It had its birth in 1971 at a meeting in Cold Spring Harbor on Long Island in New York City. And the former director of the PDB, Helen Berman, was present at this meeting in 1971 when the structural biology community came together, recognizing how challenging the science was and recognizing the importance of data sharing, took what at that time were just 7 3-dimensional atomic level structures of proteins and established this open access digital resource. So this was before anybody even knew what open access was. The data were distributed on punch cards and magnetic tape, something that's just completely foreign to us now. It's grown over the last 43 years to be a single global archive of 3D structures of biological macromolecules with more than 100,000 entries. This is a global organization. The US PDB is the RCSB Protein Data Bank. It's headquartered here at Rutgers and at UCSD, UC San Diego, funded collectively by the NSF, the NIH, and the Department of Energy. And this is part of a worldwide organization that Professor Berman established, the Worldwide Protein Data Bank, that encompasses a data center in the EU and a data center in Japan that together manage this archive of over 100,000 experimental structures and make these data freely available without restriction on the web. And you see on the right-hand side here some of the very first structures that appeared in the Protein Data Bank. The oxygen storage protein responsible for making your muscle cells look red, myoglobin. The oxygen carriage protein, hemoglobin, which just looks like 4 myoglobins. The first enzyme, 3D structure to be determined at Oxford, where I did my PhD, and another enzyme, ribonuclease, the structure of which was determined at Yale University shortly after the structure of lysozyme was published. So what is actually in the Protein Data Bank? This is fundamentally a repository for the atomic coordinates, knowledge of the atomic positions of proteins, DNA, RNA, and some small molecules such as drugs bound to either the protein or the nucleic acid, along with the primary experimental data that allowed the scientists to first determine the 3D atomic structures of these proteins to conclude that structure determination exercise. So not only is the output of the science there, but also the input, the primary experimental data. It's freely available to users around the world, and it is routinely used in every major drug company, every biotechnology company around the world, and used by HIV researchers and drug hunters alike every day. So here, this is what the file actually looks like. You can see the positions of the atomic coordinates of a carbon atom, an oxygen atom, additional carbon atoms, all part of the amino acid named asparagine, which is one of the 20 amino acids that makes up polymeric chains that are the proteins that make life possible. Every day, 1 million files just like this one are downloaded from the Protein Data Bank. This is an enormously important resource that has accelerated research, drug discovery, and of course is playing an important role in education. And the diversity of the holdings in this archive are spectacular. It includes the proteins that are responsible for locomotion, for muscle. This is actin and myosin. It includes viruses. It includes the proteins that sit in cell membranes that receive signals such as the neurotransmitters are all sitting in the membrane here. There are the proteins that are responsible for packaging DNA, for replicating DNA, for turning genes on and off. The molecular machines made up of protein and RNA that are responsible for decoding the messenger RNA and turning it into proteins, the ribosome. This is just a smattering of the 100,000 structures that are available in the archive. And we have a very, very diverse user community, as I've touched on. We cater to biologists of all ages and all stripes. We cater to a lot of other scientists who are not directly involved in biology, but are involved in things like the development of software, involved in discovery of drugs, a whole host of different activities. A major portion of our user population are teachers and students, and we have a special rendition of our website called PDB 101, which I'll show you some of the details of in a moment, that help introduce the archive to the students, to the teachers. There's a lot of use of the material in the archive by people in the media, and of course, there's always interest from the general public, particularly when there are holdings within the archive that are relevant to human health, such as Ebola, and I'll show you an example of that in a moment. This gentleman is probably well known to some of you in the audience. He's a professor next door. That's Joe Marco Trigiano. I told you that in the old days, everything came along on punch cards, on magnetic tape, etc. Of course, today it's all done through the web, and you can visualize our website and use that as the portal to get into the archive, either on a laptop or on a mobile device. We have a PDB mobile that you can download at no cost from the App Store, either for a Mac device or for a what's the rival of the Macs? The Microsoft, Microsoft type device. And you can search for proteins, you can visualize them, you can compare them, you can get access to a lot of external data which is linked to the contents of the archive. You can generate reports, and you can also use the resource for education, particularly if you're going through the PDB 101. Here's the web page for PDB 101. It provides a structural view of biology that's arranged in various topics: health and disease, nanotechnology, and there are a lot of different tools that you can use within the archive to actually help understand biology at molecular and atomic levels. So we have paper models, so you can see what DNA looks like. You can see what tRNA looks like. You can visualize proteins through models. There's a series of posters that you can download and print. There are animations that we make available, both animations of our own creation and those that we are collecting from other people. An important feature of the PDB 101 offering is a thing called Molecule of the Month. So this is an article, if you will, a web article. Comes out every month on a different protein, more than 180 issues, and adding to that number every month. It describes the structure and function of a protein or some kind of macromolecular machine that's important for reasons medical or social or scientific. There are links there to help you learn more about other molecules that may be related. There are interactive views, and there are suggested readings. And the one that's particularly pertinent to today's topic is the Molecule of the Month that describe the 3D structure of the capsid of HIV. This is the container in which the HIV genome the RNA sits. I'll come back to this in a moment and show it to you in a slightly more digestible form. I urge you to look at our fairly recent molecule of the month, talking about Ebola proteins, what we know about Ebola, what we don't know about Ebola, where we can get some traction in terms of vaccine design, where we can get some traction in terms of drug discovery. And if you keep an eye on our website next week, you'll see a very compelling video which animates a lot of these still images that you see in the PDB 101 article to bring home the simplicity of the design, if you will, of this important pathogen, and to help you understand which of the proteins in the pathogen are of particular interest to people trying to develop both vaccines and drugs. So let me say something now about the way in which structural data has facilitated the discovery of HIV treatments. This is something that I lived through. I started medical school just as HIV was actually described. You've seen pictures of like this on the news. These are electron micrographs of human immunodeficiency virus. It evolved from a virus we now know that infects African chimpanzees. It contains RNA in the capsid here. This is the genetic material of the virus. Surrounding the capsid is an envelope, and sticking out of that envelope are protein spikes. These spikes are important for attachment to host cells. They're also important as potential targets for antibodies that would be created from a vaccine, and I'll come on to that in a moment. So again, for reference, here's the electron micrograph of the virus. You can see the nucleocapsid, the storage vehicle for the viral genome. You can see the envelope. You can see the spikes. So what do we know about this anatomically? It's staggering what we've been able to learn in the last 30-plus years about, not quite 30 years actually, about HIV. So here's a schematic with the same pose as you can see in the electron micrograph. And there are different classes of proteins. And so let me jump first to the targets of antiviral agents. These are the enzymes that sit in the virus. There are three such three-dimensional structures in the protein data bank, and their locations are shown schematically here in this artist's rendition of the virus. There are some structural proteins here, which are color-coded in blue, and these are important for not only the structure of the capsid, but also represent potential drug discovery targets. The HIV genome is here, the RNA genome organized in the capsid, and then there's a series of accessory proteins that the virus uses to overcome some of the defense mechanisms that are present in our cells. So this three-dimensional picture of the virus has been built up as a consequence of the electron microscopy and also the X-ray crystallography that's been done to understand the components of the virus. And it's this knowledge which is the thing that has empowered drug hunters to actually discover potential agents for the treatment of HIV. You're all, I'm sure, aware of how important it is to actually to test for HIV. Anyone at risk for exposure to HIV would do well to get tested. And what does testing actually involve? Testing involves, in the first instance, looking for antibodies against these HIV spike proteins, the ones that are sticking out of the membrane here, the ones that are involved in sticking to the cell. And the tests that are used routinely in the clinic these days to determine whether or not someone has been exposed to HIV are aimed at detecting the antibodies to these surface proteins, the GP41 and the GP120 that sit in the surface of the virus here. It's possible very early in infections to actually detect the presence of the virus by looking for the viral proteins alone, such as the P24 protein. This can be detected in the blood even before the human immune system has actually mounted a response in terms of antibodies. It's important in the treatment phase of the management of HIV and AIDS to actually be able to detect the viral RNA. That's how we actually determine whether or not the treatment is being effective. We're assessing what's called viral load in the bloodstream. And the other thing that's done in terms of testing is to determine whether or not a person who's been exposed to the virus has been exposed to HIV-1 or HIV-2. And this is important because HIV-1 is considerably more pathogenic for humans than is HIV-2. So about the drug targets, these represent the Achilles heel of the virus. And what we're showing here schematically is the life cycle of the human immunodeficiency virus when it infects its target cell, which is the CD4 immune cell. The CD4 cell, CD4 positive cell, is the cell that is required to educate our immune system about foreign invaders. And the thing that makes HIV so damaging to the human immune system and sets up the possibility of the immunodeficiency that's seen in the later stages of the infection is that the virus infects these CD4 cells and kills them and depletes them from selectively from the body. So you can see the virus coming in, it interacting with the spike proteins, interact with proteins on the surface of the cell, and they get in. And then there's a step involving the dissolution of the capsid, exposing the viral RNA, and then this is reverse transcribed, as it's called, is turned into a piece of DNA, which then gets integrated into the gene, into the chromosomes of the CD4 cell. Then in the normal course of events, what happens is that gene that's now embedded in the human genome now makes RNA copies, RNA transcripts of the DNA genome. Those can then leave the nucleus, and they can set up the manufacture of the necessary viral proteins, and then ultimately the packaging of the virus and the release, the budding, and the maturation. And I'm going to tell you about two different classes of drugs, one that interferes with this step, the reverse transcription step, and the one that involves the blockade of the protease step. There are other potential entry points for treatment, such as blocking the fusion of the viral membrane with the cell membrane, blocking the entry. The timetable of work that's been done in the pharmaceutical industry and by academic scientists, one of whom is in the room with us today, Professor Eddie Arnold, achieved a signal breakthrough in 1987 when the FDA approved the first of the reverse transcriptase inhibitors. And this was actually a chemical that was literally sitting on a shelf in a pharmaceutical company for which a clinical use had not previously been found. It emerged that it was effective at blocking this step of turning the RNA genome into a DNA copy, and became the first drug approved to treat HIV. And then what you saw less than a decade later was the discovery of a set of medicines, the first of which was approved was this Invirase, which blocks the maturation step of the viruses. And this is an inhibitor of the protease that's required for viral maturation after it's left the cell. And then what's occurred since 1995 is that there's just been a host of approvals that have occurred. New reverse transcriptase inhibitors, new fusion inhibitors, and in the inhibition of the HIV integrase. This is the enzyme that's responsible for getting this piece of viral DNA into the human genome, and also the first entry blocker was approved in 2007 that blocks this step in the life cycle. So scientists, both in drug companies and in academia, have put an enormous amount of effort into actually trying to understand what these Achilles heels are of the virus, and then to use the traditional tools of drug discovery along with the structures that have come, the 3D structures that have come of these targets, to actually create a host of drugs that now allow people living with AIDS to live with AIDS as a chronic illness, greatly extending survival times over what we saw in the early 1980s when I was at medical school, where there was no treatment available, and then the revolution that occurred in '87 with the appearance of Retrovir in the armamentarium. So I'm going to show you two very quick movies that show you some of the details of the reverse transcriptase and the two types of reverse transcriptase inhibitors. Before I show you the movie, I'll just explain that the Retrovir, the very first one of these drugs, actually binds directly in the place on the surface of the virus where the synthesis of the DNA copy of the RNA, the virus RNA genome occurs. This is, and you'll see this in a little bit more detail in a moment. And then you'll see in the second movie how a drug such as nevirapine binds to a different place on the surface of the protein, effectively blocking the action of this DNA synthesizing machine, but doing so at a distance instead of right at the heart of the active site. Both of the structures I'm going to show you were determined here at Rutgers by Kalyan Das and co-workers in Professor Arnold's lab. They represent important steps in our understanding of how these drugs work, why they work, and what might be done in the future to actually make even better drugs. So let me show you the first of these videos. So when reverse transcriptase works, it's actually two proteins coming together. They're binding this RNA-DNA hybrid here, and then right in the active site of the enzyme, you can see the drug AZT. This is similar but different from what you see with nevirapine, where, again, you've got the two proteins coming together, interacting with the RNA-DNA hybrid here, the double-stranded nucleic acid. And you can see that the way that nevirapine and its cousins, the other non-nucleoside reverse transcriptases, actually work is by binding to a site which is actually distant from where the action is in the virus. But that sets up a disruptive structural change which blocks the production of DNA from the viral RNA in the active site. So two drugs working in different ways. This is important, as you'll see in a moment, to help overcome the possibility of drug resistance. If you use only one drug, a mutation here that allows the enzyme to work no longer binds the drug, suddenly you've got a resistant strain of virus. But if you're giving this drug and this drug simultaneously, the likelihood of a mutation here and a mutation here occurring in the same gene is very, very remote. And it's this combination strategy that is at the heart of the way in which we're now able to manage HIV/AIDS as a chronic disease. Another important player in this, as I mentioned earlier, are the so-called protease inhibitors, the first of which to be approved was sequanavir. So it binds to it binds in the active site of the protease enzyme, which is responsible for virus maturation. If the virus is unable to mature, it's not infectious. So you simply short-circuit the action of the virus, by blocking this final step in the life cycle. So again, we're looking at two copies of the protein, two copies of the HIV protease, and right in the center here, where the action is at the active site, you can see the way in which the yellow atomic stick figure drug clogs up the active site, clogs it up in a way that it's unable to work the way it's required for viral maturation. So you just saw the top-down view. This is the front view. These two amino acid side chains, these two aspartates, are where all the action is, and they're effectively clogged up. So this would be like taking bubble gum and sticking it into a lock. You can't get the key in now. You can't open the door. You can't make the virus infectious. So I've already explained to you the logic behind using multiple drugs against HIV. We can block the virus at multiple points in its life cycle, taking advantage of these multiple Achilles heel that we see in the drug. The clinical name for this approach is combination antiretroviral therapy, or CART, C-A-R-T. And it's the use of multiple drugs that allows us to reduce, in people living with AIDS, the viral load. Once you've reduced the amount of virus that's in the body, the CD4 cells can rebound, they can recover, and then your immune system comes back. By using multiple drugs at the same time, you can greatly decrease the likelihood of the emergence of resistance. One of the challenges that was seen early on when there was just AZT available was that the virus quickly mutated, overcame the impact of AZT, and then the virus was actually able to escape treatment and continue to proliferate in the patients. So the CART treatments, as they're called today, use drug combinations of 3 different HIV antiretroviral agents, and they come typically from 2 different classes. So you can use 2 of the nucleoside-like reverse transcriptase inhibitors with a protease inhibitor. Alternatively, you can use 2 of the nucleoside reverse transcriptase inhibitors with a non-nucleoside reverse transcriptase inhibitor, like that nevirapine that I showed you that bound over on the right at a distance. It's important to realize that this isn't the only way in which these anti-HIV drugs are being used. There's a practice now of identifying individuals who are not infected with HIV but are at high risk for becoming infected. So sex workers, for example, represent one such class. People living with a partner who has who has HIV/AIDS. It's routine in these cases to offer the individuals access to 2 drugs at a time that they would take daily to prevent the HIV infection. Alternatively, in the healthcare industry, you see people who end up with needle stick injuries, where they may have been exposed to HIV, and there's an opportunity in that case for post-exposure prophylaxis, so-called PEP, as opposed to PrEP, in which the individuals who've undergone the needle stick injury are treated for 28 days with 2 to 3 antiretroviral agents every day after what constitutes a high exposure risk to HIV. So this would be occupational in the context of individuals working in the healthcare industry, etc. It also can be non-occupational exposure, such as people who are the victims of sexual assault. All of these represent very important elements of the way in which the drugs are approved. You'll hear more from our colleagues from the School of Nursing about the way in which these drugs are also being used to prevent maternal to fetal transmission of the virus at prior to birth and at the point of delivery, when the mother goes into labor. And they'll tell you more about that. It's one of the great success stories of the entire HIV saga, because in the modern setting, when people get adequate medical care, adequate prenatal care, you can effectively eliminate or nearly eliminate the transmission of the virus from the mother to the baby. The real solution, of course, is a vaccine. There are no US FDA-approved vaccines on the market today. And the reason for that is not because the drug companies are corrupt and want your money to sell the pills. And it's not because people who work in drug companies are stupid. I know that's a view of some in the NIH and other places and in academia. I've been in academia, I've been in the drug industry, and I can assure you the people who work in the drug industry are every bit as committed to a cure as possible. The reason that we don't yet have a vaccine is because there are a whole host of challenges. The first is that HIV, as I told you, is an enveloped virus, and those are more challenging to vaccinate against than non-enveloped viruses. The protein spike, the GP120, is covered with sugars. So this is a 3D structure, a rendition of the 3D structure of the GP120. And so the atoms corresponding to the protein are shown here in yellow. The atoms shown here in this sort of orangey color are the sugar molecules that actually decorate the spike protein. The sugars tend to interfere with binding of anti-HIV antibodies that would otherwise be neutralizing by the immune system. So here's, in fact, a 3D structure of a fragment of an antibody recognizing part of the surface of GP120. And you can see the surface is very crowded with these sugars, which tends to block access by these antibodies. If you look at the viruses themselves, there are very few of these spikes on any given virus. And that's another problem. There just aren't very many targets for the vaccine. And then finally, to make matters even more challenging, is that we know that the gene that encodes this GP120 and the amino acids making up the GP120 protein vary among different HIV subtypes. You'll hear those referred to sometimes as clades. So there's an enormous amount of microscopic, but nonetheless very important genetic variation in GP120, which will further complicate the design and development of a vaccine. But a promising strategy does appear to be on the horizon, and this is what's been termed retrovaccinology. Over the years, it's been possible to identify that there are certain antibodies that are produced naturally by people against GP120 that really are very, very effective at recognizing, binding to, and ensuring and targeting the virus for destruction. So, armed with the knowledge that there are such things, what people are trying to do is to essentially design the virus backwards. They're working to actually identify the right immunogens, which will create these broadly neutralizing antibodies. And then the goal will be to put these immunogens together as a cocktail in a vaccine, and then administer that cocktail first to volunteers, of course, in a clinical trial after appropriate animal safety and efficacy testing, and then ultimately, of course, to populations at risk. And then in time, I hope that everyone in this room lives to see this possibility. In time, vaccination against HIV will become routine the world over, just as was the case with smallpox, where it became possible to eradicate smallpox. So I'm going to stop here and hand the floor over to my colleague, Professor Shuchismita, who will tell you about the work that she and others have been doing in developing a curriculum for HIV/AIDS that we're aiming primarily at high school students, although there's no reason that it won't be used more broadly.
So I'm going to shift gears and step away from the PDB a little bit and come back to it later. But when we talk about why do we need to learn about HIV and AIDS, this recent statistics that was reported actually alarmed me quite a bit. I mean, yes, we know that there is all this body of knowledge, and now we know how to treat and how to help HIV infected individuals to live a reasonably long life. But today, in this day and age, there are still 50,000 new HIV infections that are happening in the United States, and that's quite appalling. Even more appalling is this statistics right here, that 26% of this 50,000 new infections are in this age range, 13 to 24. So these, if you think about it, these are school-going, college-going kids. And so the question that I started asking is that why isn't that the education system addressing this? Why aren't the high school teachers and the college teachers really working at this point and saying, like, do you really understand what is going on? Do you really understand the science? And I had a conversation with some of the teachers that I was working with, the science teachers, and I said, are you talking about HIV in your classroom? And they said no. And I said, why is that? So their response to that was, well, the health education teachers are talking about it, so we don't have to talk about it. And I said, what does that mean? The health education teachers do not talk about science. You are talking about transcription, translation, DNA, proteins, enzymes, but why aren't you using HIV as an example so that you can really make an impact and help them understand what HIV is all about, so that instead of teachers or health educators telling them do this, don't do that, they can make a choice for themselves as to what they should or should not be doing. And so this started this whole thinking. So when about 2 years ago, and in the past 2 years, we have developed various educational materials, whether it's a poster, animations, paper models, and things like that. But and I'll tell you more about how some of these got created. What we did in a more focused way was we decided that we are going to focus our outreach efforts on HIV and AIDS to maximize the use of our resource, the data that is available that the scientists were using to develop and design new treatments for HIV. And then hopefully this would all come together to something that will be meaningful for the students. So what we did is we created a program where we had teacher workshops. We started with the students and we went on to the teachers. We had public symposia. Last year we had another World AIDS Day symposium like this. And then when we started talking to the teachers and asking, like, is this really helpful? Their answer was that, yes, this is great. We love the resources, but we really need a new curriculum. This was what the teachers' feedback was. And so what we decided after doing this for a year is that maybe we should just get together with the teachers and come up with a curriculum. And that's what we did this past summer. So 5 New Jersey high school teachers, one of whom is sitting in the audience here, came together, and in this very room, we spent a whole week learning and designing lesson plans and activities that were, not just the teachers themselves. We had educators, scientists, clinicians, and various experts from the Rutgers community come and talk to these teachers. The teachers would learn in the morning, and in the afternoons they would spend time developing lesson plans that we've put together. And at the very first discussion session when we had this, the teacher said that, well, we can't come up with a curriculum that will work for everybody, because there are the AP, the advanced placement biology students, who are learning about molecules and atoms and proteins and so on and so forth. So some of the material that we talked about, the videos that we saw, we could share with them, they would understand. But then there are biology classes at the 9th and 10th grade level where they barely know what a protein is. They know there are some things that are there in the cell. So to tell them that this protein has a mutation here, it's too much for them to digest. So we had to develop another level of this curriculum that was appropriate for their understanding and for them to digest. And so we did come up with 2 levels. So these 2 curricula were developed. And the focus here was purely science. We strictly refrained from any behavioral discussion. We said, we just want to understand what is the science, what is happening in the virus, what is happening in the body, what are the immune cells doing before exposure, during exposure, after exposure, how is the body responding to these different drugs, how can we understand whether these drugs are working, what are the clues. So we strictly stayed with the science and with the molecules. And we also made sure that these curricula, these lesson plans that were developed were aligned to the various standards that the teachers are held to. Because the teachers, they said that, for us, the major challenge is to cover the core curriculum and the next generation science standards and the AP learning objectives. And we don't have time to come up with these new lesson plans and ideas. So we want it all ready and, ready to go. And this is what we did. So a first draft of this is now available for testing. It's on the public website at this location. So if you know of a high school student or a high school teacher, I would greatly appreciate it if you could spread the word. We have these flyers out here in the front of the room that you're welcome to take and share it with your friends and family. The other thing that we did to engage the youth was today, nowadays, everybody, all at least youth, the teenagers, are kind of stuck to their cell phone, to their tablet, to their iPad, to their whatnot, and they're watching videos. You ask them, like, how do you do this? Oh, let me go to YouTube and figure it out. So that's how they think. So we said, like, well, if that's the medium for their learning, why don't we actually engage them in creating videos like this? And we had a video challenge last spring in 2014 where we said that we challenged the students to come up with any aspect of HIV and AIDS and tell us a story, a molecular story, in 2 minutes or less. So this was the very first time we were doing it. We had no idea what response we would get. We got 23 contestants, 23 entries. They were actually not just from New Jersey. We were pleasantly surprised to see participation from Ohio, from Massachusetts, from Pennsylvania, New York. And we had some really esteemed judges who looked at these videos and they provided their feedback and commentary. And many of the judges, or some of the judges, are here in this room. And then the videos are given some awards. Now, this is an approach that we are learning, and I guess the schools are also getting used to this idea. And so now the teachers say that, oh, this is a perfect project to do after the AP exam. I said, however you want to fit it into your school year, that's great. And this year, this upcoming year, our video challenge is detecting and combating HIV. So this time we are doing, again, on the HIV/AIDS theme, but it's a more focused theme. And the hope is that the students can not only, create a video, but also really recognize the fact that testing is important, that, that there are these medications and treatment options that are available, and for them to hopefully understand how these things work. And so there are flyers about this over there too. Again, if you have friends, family members, people you know who might be in this group, please share this, and hopefully we will get a good response this year. The details will be announced in January. So now, in saying all this, just to summarize, you heard from Dr. Burley about the protein data bank. You heard a little bit about how the treatments for HIV work at a molecular level. And I hope that you can take this understanding and share it with others and use it to think about what the PDB can offer, what this field has accomplished in progressing this far. And through these efforts that we are conducting, whether it's the curriculum or the HIV challenge, I hope you will be able to share and be part of this excitement in empowering just not you, but others in the community. So I'd like to close by acknowledging our whole RCSB PDB group. So this picture was taken in a little celebration that we had to mark the 100,000th structures in the PDB. And it spans the team here at Rutgers and a colleague in Colorado, the team in UCSD. And this is who we are. I'm happy to answer questions. I'm sure if there are questions for Dr. Burley, we'd be happy to answer any questions. Thank you.